So that brings us to genetic expression. So we talked about how DNA replicates, and that's before mitosis and meiosis. Now we're getting into genetic expression. Okay, so genetic expression is using your DNA to make proteins, and when you make a protein, that expresses your genes on the outside. So it allows you, when you make proteins, it allows you to express or see your genes on the outside. So briefly, I want to talk to you about genetic expression. We've mentioned breast cancer several times and how 20% of breast cancer cases are caused by a genetic factor, okay, the BRCA1 or BRCA2 gene. Okay, so you are born with those genes. When you're born, you either have them or you don't. Hopefully you don't. But if you do, you don't have breast cancer when you're born. You don't get breast cancer until those genes are expressed. So you don't get breast cancer until one of these genes is expressed. Okay? So how it's expressed is maybe your body's under extra stress. Maybe you were exposed to extra environmental factors or some sort of carcinogen, and then your body became under stress and your body expressed that gene by making a protein. So one of those genes becomes expressed when you make the protein. Okay, so when you make these proteins, that's how genes are expressed. Now, some genes are expressed at birth, like your eye color, for example, or hair color. Actually, eye color is something that's developed over time. You don't typically know the eye color at birth because that gene isn't started to being expressed yet. But like hair color, you might have heard of babies that, were blonde hair and then their hair turned dark brown maybe after a few years. Anyway, so, and that all depends on when genes are expressed. Like I said, some genes are going to be expressed earlier than others. So, there's 2 steps to genetic expression, and these do occur in alphabetical order. Transcription, and then translation. So C comes before L. So transcription is going to be first, then you have translation. Okay, so transcription is when DNA makes RNA, just simply put. So you make an RNA copy from DNA. Okay, and then, of course, everything starts with DNA. So, of course, transcription starts with DNA, and you make an RNA copy. And it's not going to be the whole piece of DNA. It's going to be only the piece of DNA where you need to express that particular gene. Then translation occurs, and that's where all the RNAs work together to put together your amino acids. And if you remember, amino acids are the monomer of a protein. Okay, so in translation, all the RNAs work together to make the protein by putting together the different amino acids that go for that protein, and they get the instructions from the DNA. So the DNA provides the instructions for which amino acids go into the protein and what order did they go in. Okay, so one thing you might hear is the central dogma of molecular biology, and that is just simply stating that information goes from DNA to RNA to proteins, but it cannot be reversed. Okay, so I take DNA and then I make RNA, and that RNA puts together amino acids, and then those amino acids make up the protein. Can't go backwards. Okay, I can't take a protein and then make amino acids and then make RNA from the amino acids and then make DNA from RNA. It doesn't work like that. It's not possible. Okay. So the central dogma of molecular biology is basically talking about these steps, going from DNA to RNA to amino acids to protein, and they cannot, once you make that protein, you cannot reverse it. Now, can I break a protein down into amino acids? Yes, you can, but you can't take amino acids and make RNA from them, nor can you take RNA and make DNA from it. Not in a typical sense. We did talk about retroviruses, but viruses, of course, are not cells either. and it's not going to take protein and go backwards for the whole process. So transcription. Transcription, like I said, is making RNA from DNA. Specifically, it's going to be mRNA, which is messenger RNA. Now, DNA is in the nucleus. We know this. DNA is in the nucleus. DNA is also much larger than RNA because usually RNA is only going to be one particular gene that you need. It's not going to transcribe the entire DNA molecule. It's only going to transcribe just the gene that it needs at that time. So when you think of transcription or transcribe, you should think about like copying. Okay, so my mother-in-law, she does medical transcription. So what she does is a doctor will talk. A doctor will record, patient's name is blah, 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 and his birthday is blah, 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 and they're experiencing blah, blah, blah symptoms, and I'm going to run blah, blah, blah test, and I am possibly the patient has blah, blah, blah disease, pending test results. And so what my mother-in-law does is she listens to that doctor and she types out or she copies what the doctor says. Okay, so the doctor talks it and she types it up so that they can have a printed report. So that's transcription. You're copying something. Okay, if I ask you to transcribe something, I might be just asking you to copy something down. So that's what's happening here. DNA is copying the message onto RNA. So DNA is in the nucleus and it's much bigger than RNA because it has all of your genes on it. And so DNA is too big to leave the nucleus. It can't leave through the nuclear membrane through those nuclear pores that we talked about in the cell unit in order to go to the ribosomes. If you remember, ribosomes are located in the cytoplasm or in the attached to the rough ER, which is also in the cytoplasm. So DNA needs to give RNA a copy of the information so that RNA can go to the ribosome. So RNA can take the message out to the ribosome. So anyway, RNA is different from DNA in three main ways. First off, DNA is a double helix. RNA is a single helix. Okay, so double-stranded versus single-stranded. Okay, RNA, sugar, is ribose. DNA's is deoxyribose. And then RNA's 4 nitrogen bases are going to be A, U, C, and G. So instead of thymine, RNA has uracil. These are key differences, and you will see this again. Make sure you know those. So your 3 types of RNA are going to be messenger RNA. So it carries the message from DNA in the nucleus out to the cytoplasm to a ribosome. And if you remember, ribosomes are the site of protein synthesis, so that's where the protein will be made. Then you have tRNA, or transfer RNA. It brings the amino acid to the ribosomes. Okay, so somehow you have to get the amino acid to the ribosome to make the protein. You do have something called an initiator tRNA, and it has what's called a start codon on it that starts the whole protein making process. Then the last type, the third type of RNA, is rRNA, or ribosomal RNA. That is a little r, by the way. rRNA makes up about 60% of the ribosome. And remember, ribosomes are where the protein's made. So ribosomes are made of 2 things, rRNA and protein. And they are where proteins are synthesized. Okay, so this is an excellent picture to summarize protein synthesis. Okay, so we're going to start off with DNA in the nucleus. Notice the DNA is much larger than RNA. Okay, so transcription is going to be your first step. Okay, so DNA transcribes the message onto mRNA. Y'all see the mRNA here? DNA unzips just the part that it needs, so it doesn't unzip the whole DNA. It only unzips the part that it needs, the gene that it needs to express. So DNA unzips, and then some different enzymes come into play, and the RNA molecule is made. So that you will see as the RNA will have U's in it, and the DNA will have T's in it. Okay, and so then you have your RNA molecule, your mRNA. So mRNA is messenger RNA. It will then leave the nucleus and go find a ribosome. Can y'all see the shadow of the ribosome here? Okay, and it will leave the nucleus and go to the ribosome. Okay, so again, here is your RNA molecule, messenger RNA. So messenger RNA will leave the nucleus and go to the ribosome. Now, then you have your, and remember, ribosomes are made up of rRNA and protein. So then you have your tRNA that's going to come up. So what's going to happen is on this mRNA, if I have A, U, G, tRNA will have U, A, C. So tRNAs will match up its nitrogen bases to RNAs, and then it will drop off an amino acid. Okay, and then that will happen again. The next tRNA will come up. So on, So on RNA, these three letters, let's say CCC, those are called codons on RNA. And then on tRNA, those are called anticodons. So GGG would be the anticodon that goes with CCC. And this tRNA only carries one type of protein, one type of amino acid. And then once you get more than one amino acids, they will bond with a peptide bond. And this will continue until eventually you get to a code, a 3-letter tRNA that's going to be a stop codon. It's not going to code for an amino acid. It'll code for stopping the process. If you never stop the process of protein making, then you never finish making one protein. Okay, so transcription, this is just putting it all into writing. So again, DNA provides the blueprint for making proteins. If you recall, proteins are made from amino acids, and you have 20 amino acids. You only have 4 nitrogen bases. So how do you get 20 amino acids out of 4 nitrogen bases? Well, they're put into different codes, okay? So if one base codes for an amino acid, then there could only be 4 amino acids. Okay, so you have some amino acids that go with multiple codes. So you do read it in sets of 3. Okay, so a codon is 3 nitrogen bases on mRNA. Okay, so that's just 3 nitrogen bases. So there are 64 combinations. So out of 20 amino acids, and you have 64 combinations, it means some of them might have 2 or even 3 or 4 codons that work for that particular amino acid. So 61 of them code for amino acids. The other 3 are what we call stop codons, and they tell you to stop making the protein. So this is why it's called the DNA code or the genetic code. You'll see what I mean in just a minute. So in the enzyme RNA polymerase is going to go along the DNA strand, and it's going to pair up RNA nucleotides. Okay, so it's going to make up the RNA strand that way. And it is going to again go in the 5' to 3' direction. So anyway, one mRNA molecule will then transcribe the copy of DNA. Okay, so this is a picture of translation. Okay, so this is the next step. What you will get is your mRNA, which will look like this. Okay, and all they've done is they've just taken the G and brought it over, taken the C and brought it over. Okay, they're just bringing all these letters over. So this is my mRNA strand here, and if you'll notice, they grouped them into 3 letters. Each 3 letters codes for 1 amino acid. Okay, so you make the mRNA in transcription, and then in translation, you put together those amino acids. Okay, so first off, look at codon 1, GCU. Okay, so I go to my first base, which is my first letter, and that would be G. My second base would be C, and then my third letter would be U. So I have to take G, C, U. So alanine is the amino acid that goes there. I'm going to erase just to make it easier to see. So you're just finding it on the chart. So my second codon, let's do a different color, is going to be ACG. So I find my first base, A, my second base, C, and my third base, G. So A, C, G. So threonine is my my amino acid, sorry. Okay, so my third one, GAG. So G, A, G. So glutamic acid. Okay, my fourth one, CUU, C, U, U, leucine. So what I'm doing here is I am translating my RNA into amino acids. And these amino acids will then bond with a peptide bond to make a protein. So we'll go ahead and continue on here. CGG. So we have C, G, G, arginine. Just going to abbreviate that there. Okay, getting kind of messy, so I'm going to erase some. You get what I'm going at here, how I'm showing you how to do this? This is just one chart. Some of the charts have next to the amino acid, so you don't have to go through as much trouble, but some don't. So I wanted you to see it, you know, how you would see it on the test. So AGU is the next one. So A, G, U, serine. And then the last one, UAG, U, A, G, stop. So now we can stop making the protein. Okay, so we just translated. So all these amino acids will bond together with peptide bonds. And now you've made a protein. So that brings us to the written part of how translation occurs. So translation is when you make that protein, and that is also when you express the genes. So protein synthesis is the making of a protein. Okay, so translation is where you convert the nitrogen-based sequence that we just saw on mRNA into the amino acids. And then those amino acids peptide bond to make the protein. Okay, so if you take a look here, this all happens at the ribosome. Remember, the ribosomes are made up of rRNA and protein. Okay, and so protein synthesis happens at the ribosome. So you have your mRNA, that's this long strand, okay, and that mRNA is going to connect to your tRNA. Okay, there's your tRNA. Okay, hang on, let me fix that. This would be tRNA. Okay, so on mRNA, these three letters would be called the codon on mRNA. And these three letters on tRNA is called the anticodon. Okay, so the codon matches up to the anticodon. And that particular codon will always code for this particular amino acid. And that's why it's a genetic code and it's predictable. I always remember codon, mRNA, anticodon, tRNA. Make sure you know which is which. Okay, tRNA, anti has a T in it. So then, this will continue. Okay, so your next your next codon, GUG, will match up with CAC every time, the anticodon, and that will always carry this particular amino acid. And then you keep dropping off these it'll slide forward. It'll keep sliding forward on the ribosome. So if you notice, this one is not on the ribosome yet. Okay, but this one, once it drops off the amino acid, it will slide forward. So this one would have already been done. Anyway, so this will keep happening. These amino acids will peptide bond, and eventually you'll get to the stop codon, and then you have your protein. Once you have your protein, the gene can be expressed. So, again, this is just writing it all out. The first codon on mRNA will attach to the ribosome. Remember, ribosomes are made up of rRNA. So that first codon is usually a start codon. Often it is AUG, and that codes for the amino acid methionine. Okay, so that is usually the start codon. Now, ribosomes can only hold 2 tRNAs at a time, and so it has a P site and an A site. So after translation, they'll shift because it can only hold 2. It'll just keep shifting to the next codon and to the next anticodon, so that the next amino acid can be brought. Okay, so amino acids are going to be transferred by the tRNA to the P site, and then the mRNA will then go from the P site to the A site. So P will be first, then the A site. So then you get the peptide bond. Remember, I told you that your amino acids will peptide bond. So once you have an amino acid at the P site and the A site, then they'll peptide bond. That's kind of what this is saying. You don't have to focus so much on the P site and the A site. This is just giving you that information if you wanted to know it. Anyway, so when your tRNA at the A site gives up the amino acid to your protein that's in the process of forming, then it will move off into the cytoplasm. When that tRNA moves off into the cytoplasm, it doesn't have an amino acid anymore. So what it's going to do is it's going to go into the cytoplasm, and it's going to find that particular amino acid that matches to it just perfectly, the only one it can carry. It will find another one to attach to and grab it a new amino acid, and then that way, the next time the cell needs to synthesize a protein, it will have the amino acid. So the tRNA is going to drop off the amino acid and then go find another one, basically. So anyway, the order of events leading to genetic expression: DNA, then RNA, then amino acids, then protein. Okay, remember, this cannot go reversed. So protein synthesis is genetic expression. This is how you express your genes or show your genes on the outside. Okay, so this is another great picture of protein synthesis. Okay, so again, you have your ribosomes down here. Okay, usually it's just one ribosome, but anyway. So you have your ribosomes, which are made up of rRNA. Okay, and then you have your mRNA here. Okay, so maybe you have your mRNA codon CCC, and it's going to attach to your tRNA anticodon. Maybe it's GGG. Okay, and it's going to drop off an amino acid. Okay, and so it's going in this direction. So this would be the P site, and this would be the P site, and this would be the A site. So again, you have another tRNA. Okay, and so let's say this is AAA. So maybe your mRNA codon's AAA. Your tRNA would be UUU. Okay, so you get them both here. There's your peptide bond going to form between your amino acids. And then once the P in your A is going to come up. I'm sorry, I wrote those backwards. This would be the A. This one would be the A. This one would be the P. Okay, so once your A moves off, then your P can move into the A spot, and then this next one can come up. Okay, so if you look, you've got some free tRNAs and some free amino acids. So the appropriate tRNA is going to bond to the appropriate amino acid, so then it can go on to help the cell do protein synthesis. So again, you've got all three RNAs here. You got your rRNA in the ribosome, your mRNA, and then you have your tRNAs.